Hey class, in this recording we're going to focus on blood, so let's dive into it. As we look at blood, we need to talk about how there's just a lot of myths of blood. We used to think that there's this mysterious, vital force in our bodies that kept us alive. And in order to treat it when we were sick, we had to remove bad blood from our bodies for medical reasons. This bad blood was part of the field of medicine that was concerned with the four humors, the four fluids of the body. And those four fluids were saliva, bile, urine, and blood. Now it's pretty hard to remove bile and saliva from the body. Again, I guess to remove urine, you just have the person go to the bathroom. So what older or more traditional doctors in the Middle Ages, all the way up to as recently as the 1700s did, was they would bleed their patients out in order to treat their illnesses, because that was the blood that they had the most ability, or the humor that they had the most ability to remove from the body. And famously, this is what killed our first president, George Washington. He was ill, he had a fever, and to treat his fever, his physician bled him. And that obviously, now when we think of that, that's just crazy. I mean, when a body's stressed out, you don't want to take that organism, that human being, and physically stress them even more by having them bleed repeatedly. But people used to do that frequently, and that's what killed our first president. When we think of blood cells, these blood cells were seen in the very first microscopes that were made, which is pretty impressive when you think about it. When you think about how small those red blood cells are. And when we look at the study of blood, that is hematology. Heme referring to blood, ology referring to the study of. Oh, here we go. So as we're looking at this, our circulatory system, which moves blood around the body, is going to consist of a pump, that's the heart, the fluid itself, which is the blood, and then the tubes that's moved in, which is the blood vessels. Now, this cardiovascular system is the biggest part of our circulatory system, and other textbooks, other individuals will say that the lymphatic system is another part of our circulatory system, and that we have a third circulatory system with cerebral spinal fluid. But really, generally speaking, when you say circulatory system, most people are going to refer to the heart, the blood, and the blood vessels, with very few exceptions. And this cardiovascular system is going to refer to just the heart and the blood vessels. Cardio meaning heart, vasculature for blood vessels. So if a tissue is avascular, it is without blood vessels. If it's highly vascularized, it is going to have lots of blood vessels in it. As we look at the circulatory system, which involves moving the blood throughout the cardiovascular system, our circulatory system is going to function to transport gases, notably oxygen gas and carbon dioxide gas. We'll have various nutrients, waste products, hormonal signaling molecules, and stem cells for tissue regeneration being moved throughout our bodies. We use our circulatory system to protect ourselves. In particular, we have white blood cells, leukocytes, that are going to move around our circulatory system and limit the spread of infection, destroy microorganisms, destroy cancer cells, and we have platelets that help to initiate the clotting cascade. We also are going to have leukocytes or white blood cells aid us in inflammation and our inflammatory responses. And then finally, we use our circulatory system for regulation. We use that to help regulate the amount of fluid in our bodies, to stabilize our body's pH, and particularly the pH of the extracellular fluid, ECF, extracellular fluid, and to help even out the body temperature. So if we're overheating, we send more blood to the surface. If we're too cold, we send blood to those internal visceral organs. Now, let's talk about how much blood we have in a body. So we're looking at a quote-unquote average adult. An average adult is going to have 4 to 6 liters of blood, or 1 to 1.5 gallons of blood-ish in their body. And, if we're looking at an adult that is very large, a bariatric or morbidly obese adult, they have a larger body volume. They're going to have more blood to correspond with that larger body volume. And vice versa, if you're looking at somebody who's just a teeny tiny little stick of a person, and they weigh maybe 100 pounds soaking wet, they have a much smaller body volume, and because they have that much smaller body volume, they're going to have a much lower volume of blood for that body. As we look at blood, it's a connective tissue. and it's a connective tissue that's a liquid at room temperature. Its extracellular matrix has almost no protein fibers in it. We have the plasma. Blood plasma is the extracellular matrix of our blood. And it's going to be a clear to light yellow fluid depending on the hydration status and physiological status of the individual you collected that from. And then in addition to the matrix, the part the non-living part of the tissue that's outside of the cells, we have the cells themselves, the formed elements. So we have the blood cells and the cell fragments. And when we think of cell fragments, we typically think of platelets. When we think of blood cells, we think of the red blood cells, also known as the erythrocytes, and the white blood cells, also known as the leukocytes. So as we look at these erythrocytes, I really love this pic right here. It really helps to illustrate that biconcave shape or the biconcave nature of a fully developed erythrocyte. And we can see those erythrocytes down here in that photomicrograph. A erythrocyte is also known as a red blood cell. Next we have a thrombocyte, also known as a platelet. And we can even see thrombocytes like right here, for instance. That's a thrombocyte. Now, when you're looking at blood histology or blood on a microscope, we're always going to use these red blood cells as a frame of reference. So thrombocytes are smaller, much smaller than the red blood cell. And then here we have a leukocyte, a white blood cell. That leukocyte, generally speaking, will be bigger than a red blood cell. Now, when we think of thrombocytes or platelets, they are fragmented cells that come from other cells within our bone marrow. The special cell within our bone marrow is called a megakaryocyte, and that megakaryocyte is going to emit fragments of itself into the bloodstream, and those fragments become thrombocytes or platelets. When we think of our leukocytes, also known as our white blood cells, leuko for white, cyte for cell, our leukocytes are broken up into 5 categories: the basophil, eosinophil, neutrophil, monocyte, and lymphocyte. Those 5 different leukocytes are broken up into 2 broader categories based on the presence of granules or no granules. So as we're looking at this slide, the 3 leukocytes that have granules are referred to as the granule sites, and they are shown here. Notice how all 3 of these granule sites have lots of speckles within their cytoplasm. And then the agranulocytes, and this got kind of cut off here. This is supposed to say monocyte right here on this slide. We have lymphocytes and we have monocytes. These agranulocytes are shown over here. We have the monocyte and that lymphocyte. Notice particularly as we look at these, with their extracellular matrix, the monocyte has a lot of clear extracellular matrix, and the lymphocyte has hardly any clear extracellular matrix. Another way to think of this is that the nucleus of a lymphocyte takes up the majority of the cell's volume. Now, I included this funny clip art picture here of a monkey eating bananas, because if you can remember the rhyme, "Never let monkeys eat bananas," you can remember the most common leukocyte to the rarest leukocyte in order of most common to least common. So, never let monkeys eat bananas stands for neutrophil, lymphocyte, monocyte, eosinophil, basophil. So never let monkeys eat bananas. Which means, as we look at this, that simple mnemonic, neutrophils are the most common cell type, and then basophils are the rarest leukocyte, the rarest kind of white blood cell within our bodies. Now, when we look at our blood as a whole, our blood can be analyzed when a hematocrit is performed. Traditionally, to perform hematocrit, we collect some blood from an individual, and then that blood is spun down in a centrifuge. This used to be a common high school lab activity up until the 1980s, and the HIV epidemic, or I should say pandemic, which is endemic now, became a reality of our world. And what we do is we take that blood and put it in a centrifuge and then separate its components. We typically are going to have the red blood cells go to the bottom of the tube. We have the white blood cells forming the buffy coat in the middle, and then we have the blood plasma, or the extracellular matrix, making up the top component of this. This process of spinning down blood to perform a hematocrit is now rather outdated technology. Typically, most hematocrits are performed using a form of spectroscopy, where just a laser beam is shot through the light, and based on the amount of light going in and the amount of light coming out, we can determine the properties of that blood. So we'll just talk about hematocrits using this old school technique because it's a little bit easier to understand. As we look at a hematocrit, erythrocytes, red blood cells, are going to make up between 37 and 52% of the total volume. Generally speaking, if we were to really just average it out, we typically think 45% of whole blood is erythrocytes. And then 55% is blood plasma, and then less than 1% is going to be the white blood cells and platelets combined. So these numbers aren't always going to exactly add up to 100% because we're rounding them down to, in some cases, 1 sig fig or 2 sig figs. And we're trying to go to the nearest whole number. So the math is a little bit off. So as we're looking at the buffy coat, that buffy coat is going to be made up of the white blood cells and platelets, and then the blood plasma is going to have mostly water. We'll have proteins, nutrients, dissolved ions, aka electrolytes, waste products, hormones, and blood gases. So when somebody is donating blood plasma, they will typically have blood drawn out of their body. The blood is spun down, and then the erythrocytes, or really the formed elements, are going to be put back into their body, and then will be diluted with a saline solution while they're pumped back into the body. And then the blood plasma is saved and given to patients that need a plasma transfusion. That's all we have for this recording. If you have any questions or comments for me, please feel free to post them on the class discussion board or to shoot me an email. And as always, happy studies.